Hello, I'm going to talk about how urban contamination might be, as a stressor, affecting adaptation between duckweed and their microbes. I think that the speakers so far have probably convinced us that adaptation produces trait differences, including adaptation response to abiotic and biotic stressors. So I'm not going to spend time introducing that. We'll just jump right into what is duckweed. It's Lemna minor. It lives all across North America, and that includes ponds in urban and rural areas. It's a small floating aquatic plant and reproduces clonally. You can't see individuals in either of those pictures, but that green film on the water is duckweed. It hosts a variety of microbes, like plants, on its tissues, hanging off the leaf, around the roots. And these span a wide diversity from bacteria to fungi, just like terrestrial plant microbiomes. And also like terrestrial plant microbes, these appear to be facultative interactions from the perspective of the plant, but on the large part beneficial. However, different from terrestrial plant microbiomes, there appears to be fewer taxa, on the order of dozens rather than thousands of taxa. And they're more culturable. Duckweed is like a floating plate of agar media. And so most of those microbes that grow in duckweed also grow in the lab easily. So we collected duckweed and associated microbiomes from a number of ponds around the Greater Toronto area. The city center is marked by that little star. And these ponds range from urban to suburban and rural environments. We collected the duckweed along with their associated microbes, and then separated the two in the lab. Among the contaminants that we might expect to vary along this urban to rural gradient, one of the major ones is zinc. Zinc leaches out from roofing materials, especially gutters, and also is a major component of tire dust. So when the tires wear down, all of that dust is on the road. There's a rain event, all of it washes into the nearby water systems. There are sources of zinc in rural areas, especially, and sometimes in agricultural fields, they'll use it to balance fertilizer ratios. So what I think about is what's happening when plants are responding to both biotic and abiotic sources of fitness variation, and will variation in an abiotic stressor along this urban-rural contamination gradient affect adaptation between duckweed and their microbes. I'm also going to talk about how adaptation might be altered by the presence of contaminants. So do we see alteration in the tolerance of contaminants when there is local adaptation? And then finally, sort of sum up with how these different microbes and how zinc contamination might be together or separately affecting duckweed traits. So first, there's a lot of different definitions of local adaptation. I just want to show you what I'm referring to when I talk about it. So when we're growing a plant, the duckweed, in different microbe environments, I'm comparing the average of the matched plants and microbes from the same sites to the average of all unmatched comparisons. And so matched I would call sympatric, and unmatched I'll call allopatric. And the experimental design is for each of these 10 sites, there are duckweed and microbes, and I'm recombining an all-by-all comparison where there's, for each of those, there's 90 allopatric combinations and 10 sympatric combinations. I'm repeating this experiment in 2 different levels of zinc, one relevant to sort of background levels across Ontario, and another approximately 4 times that, and a level consistent with a runoff event from a highway into a stormwater pond. We grew these up in 24-well plates, where there's a single duckweed and a single microbial inoculation in each plate, and then used ImageJ to quantify the size of the duckweed at the beginning and the size of the duckweed at the end of the experiment after 14 days. And you can see that in addition to duckweed growth, that is the same well at the beginning and the end, there's also some microbial growth happening as well. Okay, so before we ask if there's variation in local adaptation, just want to show you whether or not there is any local adaptation at all. So here we're comparing the surface area of growth of duckweed that are with microbes from an allopatric site and duckweed that are growing with microbes from sympatric sites. And on average, the duckweed are growing better with microbes from their home site. And I'm just going to zoom this plot out and show you what all of the data look like instead of just the mean and the standard variation. So there's a lot of variation happening there. And we had this hypothesis for where some of this variation might come from, whether or not local adaptation is varying along this urban-rural gradient. So now I'm plotting the duckweed populations against the distance they are from the city center of Toronto, and showing you that same allopatric-sympatric contrast. And we can see that what was driving, the populations that were driving that sympatric-allopatric difference, or the signal of local adaptation, are primarily the rural populations. We also wanted to ask whether or not this local adaptation between duckweed and microbes might influence the tolerance of zinc, of contaminants. So if microbes are assisting duckweed in this, and dealing with zinc by sequestering it, by increasing their growth rate and diluting the concentration in their tissue, we'd expect to see an increase, potentially, of more local adaptation at higher zinc. And that is what we see. So the solid line is the higher zinc treatment, and the difference between allopatric and sympatric combinations is only significant in that half of the experiment when you split it out. However, what you're also seeing is that there's really no difference in growth on average across these 10 populations between the higher and lower zinc treatments. For a different set of populations, we saw a 25% decrease in growth across the two high and low zinc treatments. So I guess what this is telling us in total is that zinc contamination isn't universally stressful across duckweed populations. Some populations are stressed by it and some aren't. So we also wanted to ask whether or not microbes and zinc are affecting duckweed traits. And I'm going to start with just one trait, which is aggregation phenotype. So sometimes when the duckweed are producing daughter fronds, they stick together, and sometimes they don't. And this phenotype might affect how well the duckweed can tolerate some water flow in the environment. The more they're stuck together, the more they'll stay put. And it also has effects that are relevant for the larger ecology of the pond. As there's more duckweed stuck together, they could be more effective at shading the water, preventing warm up of the water, and keeping things cool for fish. So this change in how much things are aggregated across the experiment, or across the experimental time, might again differ between this zinc levels, and here I'm specifically asking about the sympatric effect and whether or not that's different at the different zinc environments. And in low zinc, duckweed are becoming more aggregated when they're growing with their sympatric microbes, but there's no, that effect is not there at higher zinc. So zinc might be reducing a plastic response to sympatric microbes. Whether or not this is beneficial to fitness is a little bit unclear, especially in a well plate in the lab. I want to zoom out to another trait, which is color intensity. So all of these duckweed have green tissue in them, but in some cases they're clearly more green than in other cases. So we might expect with more color intensity that these plants have higher leaf thickness, more chlorophyll content. They might have more potential to grow in the future. And now I'm showing you what that phenotype looks like across all of the different combinations. They're just so I'm here showing you the resultant phenotype, and then on the other side, the matching grid setup of the experimental combinations. On the left is the low zinc half of the experiment, and on the right now is the high zinc half of the experiment. And all you're meant to draw from this is that there are different phenotypes and different combinations, and that the same combination might not always give the same phenotype. If we take all of those little squares and plot the color intensity phenotype at low zinc against the color intensity phenotype of that combination at high zinc, I think I can show you that there's really no correlation between the two, showing a great deal of plasticity in duckweed. If I take that and show you the correlation between the high zinc and low zinc phenotype of a particular combination across various other traits that I've measured, again, we don't really see much correlation. So where this unpredictability comes from could be the duckweed or it could be the microbes. So we're going to take those, all of those treatment outcomes and average them across the duckweed populations in that extra column I've added, and average it across the different microbe sources in the extra row at the bottom. And then I'm going to compare the correlation between the duckweed average from the same population and the microbe average from the same population. And for the duckweed, they're somewhat similar, weakly, but somewhat similar across the different zinc treatments, duckweed populations that grow more and are darker green in one at low zinc are also similar at high zinc. However, for the microbial treatments, the story is different. The effects are less correlated, or when they're correlated, appear to oppose. So one interesting thing going on there is that the microbes that cause duckweed to have lots of fronds at low zinc are the ones that cause them to have the least fronds at higher zinc. So to sum all this up, as to whether or not adaptation between duckweed and microbe is varying across this urban-rural gradient, and the answer there is yes. And I just want to point out here at this moment that I can't tell you where this effect is coming from. It could be adaptation in the duckweed, or it could be changes, adaptation or community composition on the microbe side of things. At this point, there's something looking like local adaptation in the interaction, but where it's coming from is unclear. When we asked whether or not local adaptation was affecting zinc tolerance, it's pretty clear at this point that the microbial community that duckweed is associating with is more important for fitness than the contamination environment. But that environment did alter whether or not we were detecting local adaptation. So local adaptation is contingent on the environment. And we did see a lot of dependence and codependence on microbe effects on duckweed traits. So the effect of a microbe or zinc contamination on the trait expression and fitness in duckweed depended on the other factor as well. And so I guess as a big sum-up message, the effects of adaptation to biotic interactions will depend on past and contemporary abiotic and biotic context. And with that, I'd like to acknowledge my lab and sources of funding at Toronto. So thank you.